Hi everyone, welcome to a new topic that we are going to look into today is virulence factors. Virulence factors have an important role in breaching the host defense. So to start off with, what is virulence? Virulence is nothing but the ability of a microbial strain to cause a disease. Example, pathogenic species of Mycobacterium tuberculosis and polio contain varying degree of virulence. Also, avirulent strains of vaccines are also some of the examples that can breach a host defense. Now, the factors which are going to be helpful in determining the virulence are adhesion, invasiveness, colonization, toxigenicity, enzymes, infective dose, antiphagocytic factors, communicability, route of infection, etc. Firstly, what is adhesion? It is the microbial attachment, such as bacteria, to the surface of the host. It is specific reaction between receptors on host cell and adhesive structures of bacteria, also called as adhesin, which are nothing but the pili or a frimbi. Adhesins are generally made up of proteins which are antigenic that induces the immune response. Apart from flagella and pili, other structures such as slime layer, glycocalyx, lipopolysaccharides also play a role in adhesion. Specific immunization with adhesion can be a method of prophylaxis or treatment in infections such as diarrhea in calves or in treatment of gonorrhea in humans. Invasiveness, the ability of a pathogen to spread in host tissue and establishment of infection. Here, highly invasive as well as less invasive pathogens are involved. Highly invasive pathogens cause generalized lesions. Examples are streptococcal septicemia following wound infection, while the less infective pathogen causes the localized lesions. S. aureus is the example. Once there is a surface attachment done, microbial invasion into the tissues and organs is accompanied by the disruption of the mucosal lining or the skin. Now, colonization. It is nothing but the multiplication of the pathogenic microbes wherein toxins are produced. During this stage, pathogens compete with normal flora for their nutrients, and the host begins to show the signs of septicemia, infection of the blood. Now, the production of siderophores by pathogenic bacteria is considered as an important virulence mechanism, while also bacteria, when they adhere to the surfaces in aqueous environment, begin to excrete certain slimy glue-like substances that anchor them to all kinds of materials, which is nothing but the biofilms. Nextly, toxigenicity, the ability of a microorganism to produce a toxin which contributes to the disease development. Two types of toxins are produced by the bacteria: exotoxin and endotoxin. Exotoxins are nothing but heat liable proteins which are actively secreted by the cell's extracellular. They are highly antigenic and are active in very minimal dose. They are mainly secreted by gram-positive bacteria, while the endotoxins are heat-stable lipopolysaccharides. They are not secreted extracellular but only released by disintegration. Here, we need to know that they are poorly antigenic and they are active in large doses. These are produced by gram-negative bacteria majorly. Infective dose, adequate number of bacteria should gain entry in host to cause successful infection, which is estimated by minimum inhibited or minimum infecting dose or minimum lethal dose. What is MLD? It is the minimum number of bacteria required to produce death in susceptible animal under standard conditions. Example LD50, while MID, minimum infecting dose, is the number of bacteria required to produce clinical evidence of the disease. Nextly, the enzymes as virulence factors. Bacteria produce various enzymes which directly damage the host. Some of the enzymes that we are going to look uh, here are proteases, which breaks the antibody IgA that protects our mucosal surface. Hyaluronidase breaks down hyaluronic acid, while coagulase cause deposition of fibrin around the bacteria. Hemolysin lies out the RBCs. Collagenase breaks the collagen in connective tissue. Lecithinase destroys phosphatidylcholine in the cell membrane. Antiphagocytic factors. They are of three kinds: cell wall proteins, cytotoxins, surface antigens. Cell wall proteins are of two: protein A and protein M, while cytotoxins are hemolysin, leukocidin. The surface antigens involved here are V antigen, which are seen in Salmonella typhi, while K antigen is seen in E. coli. Now, the genetic factors as virulence factor. Enterotoxin produced by E. coli and Staph aureus. Here, the gene coding for some virulence character may be plasmid borne. The R plasmid increases the severity of the disease by resistance to antibiotic therapy. The bacteriophage-directed virulence is seen in diphtheria bacilli. Now, the last two factors, route of infection and communicability. What is route of infection? See, some bacteria can only survive when introduced by their optimal route. Example, Vibrio cholera. Vibrio cholera, which are infective only when passed orally. Okay. Another case here. Different bacteria causes damage to different organs in different species. What is it? Tuberculosis bacilli. Same bacteria causes lesions in kidney in rabbits, while in gyna pigs it causes lesions in liver and spleen. Communicability, the word itself says, the ability of a parasite to spread from one host to another, which determines the survival and distribution of it in particular community. So hope you guys understood. Subscribe to our channel Dream Incubates for the videos and the updated lectures. Please like the video.